Hello, everybody. Week 3, Chapter 3. Why I love this topic? Because it helps you dive into a couple of things. To better understand the diversity of life on the planet, we have to understand their characteristics. And also, to understand diversity on the planet, we have to understand the traits of these organisms that have given them the edge, right? So, natural selection and evolution have selected for the features that we see in these organisms today. And these organisms we see today are the ones that won. They are the ones that were most successful, or they are the ancestors of those that were most successful. So let's get started. All right. So you might remember we had 3 domains of life. You're not going to forget this after like the 4th week of this class. 2 of our domains, the bacteria and the archaea, are of the prokaryotic organisms. So prokaryotic cells appeared in the fossil record kind of as we know them today-ish, about 3.5 billion years ago. Okay. And so when we take a look at those cells, we see that they're essentially a cell. They have a plasma membrane, they have the cytoplasm or the cellular goo, and then they have DNA. But the DNA is not packaged in a double membrane bound organelle like they are with the eukaryotes. So yes, they have DNA, and their DNA is going to be different. Their DNA is a circular, double-stranded DNA, and it doesn't have complex proteins we call histones. Histones help with DNA replication in our eukaryotes. So the second bullet here also says, okay, not only do we not have that double membrane bound nucleus, we don't have membrane bound organelles. The term organelle means tiny organ. So there's no Golgi apparatus, there's no chloroplast, no mitochondrion, no endoplasmic reticulum. Are there subcellular structures that help? Yes, there's subcellular structures that help with things like the ribosomes, but the ribosomes are not associated with the endoplasmic reticulum. Okay, next up, there are cell walls not only in bacteria, but in fungi and in plants. But we know that the cell wall, why am I getting all these things right now? 1 second. Okay. We know that the cell wall of our bacteria and archaea have this complex structure. And the complex structure really is going to help us understand how are they living their lives and being successful in the environments that they're in. So they have this peptidoglycan layer you're going to hear more about later, and they have some other structures that are associated with that, and it's going to help us divide up our bacteria. So as we're learning the diversity and we're starting to classify them and subgroup them, these are going to be some characteristics that are important. And the last thing there, division of cells by binary fission. So you all have probably heard of mitosis, and we don't have mitosis with our archaea and our bacteria. Instead, we have binary fission, which is a very simple way of replicating that circular piece of DNA and splitting it, and the rest of the cell gets split as well. So very simple. There are multiple phases that you've learned before, just pure and simple. So moving on, I may just pause this for a minute, make sure my microphone is working. So give me 1 moment. Okay, brilliant. My recording is working. So let's take a look at the bacteria cell structure. So you're going to see on the left-hand side the 5 bullets. So all bacteria have these features. So it shouldn't be surprising. We just covered most of those. The one thing I did not mention was the cytoskeleton. The cytoskeleton isn't really unique to bacteria, but what we know about the cytoskeleton is it's giving support to the cell from the inside out. So just like you have a skeleton that's supporting you from the inside out. So cytoskeletons support the cell. So bacterial cells do in fact have those. And then you see down below, most bacterial cells will have a cell wall and a surface coating called a glycocalyx. And the glycocalyx can come in 2 varieties, and so we will break that down in just a little bit. Let me move my face over here. So if you take a look, I love this figure because it's reinforcing through pictures what I just said. So in all bacteria, you have the things in the purple color. On the left-hand side, at the top it says in all bacteria, and then on the right side, in some bacteria. So these are some structures that we are going to spend a little bit of time on. If we don't spend time on it, then you don't need to know it. It's just like extra stuff to understand. Okay, for those of you that are visual learners, you might want to pause on this slide because it's going to help you remember kind of the layers from the inside out of the bacterial cell, which I find very helpful because I'm also a visual learner. And then it helps you kind of subcategorize the structures. But we're going to spend some time going over these structures now. I just want to make sure that you can pause here if you want to look at this. This picture is also in your book. It's the supplemental figure from the authors of the book that is going through the bacterial cell from the internal, the boundary, and the external parts of the cell. Okay. All right, so let's take a look. This is a picture of a bacterium. It's not in your book. It just looks different than the one that you saw before, but I like it because it simplifies it. So I will try to talk you through the figure as well as the structures and their functions. Okay, let's start with flagella. So you might know flagella best when we talk about sperm, right? The flagellum is helping that singular sperm swim to its appropriate destination. So we know that there's not just one singular sperm that's involved in sexual reproduction, but that flagellum is helping push along that sperm cell. Some flagella, actually most flagella, will pull an organism through as opposed to pushing it. So you see that this little bacterial cell has one really long flagellum, and they tell us that the flagellum has more of a whiplash kind of movement that helps it pull that cell through the environment. All right, then we have pili and we have fimbriae. So I want to next go over the fimbriae. Fimbriae are like rigid bristles that help the bacterium stick to the surface of where it's living. Now, if it's pathogenic, it's probably trying to stick to the surface of a cell that it wants to invade, right? If it's living in the environment, the fimbriae is going to help a singular bacterium cell essentially kind of interconnect or attach to other bacterial cells. And this is going to help them essentially live in bigger groups. You might have heard the phrase safety in numbers. So bacteria have an edge on us in that they can hang out with others in clusters and essentially form more protective layers. So it's harder to treat them with antibiotics. It's hard to remove them with disinfectants. So we'll get more to that. The pili. Pili is kind of interesting. These little projections that go from one bacterium to another, and they exchange a copy of that circular DNA. And this is how bacteria do what I call bacterial sex, which is called conjugation. Okay, so inside out. So in the middle of this cell, let me get my little cursor here, you see essentially this big purple wad of the circular chromosome. This is what we call the nucleoid. So it's the region of the cell that has this densely contained, if you will. It's not really contained, but compacted DNA. So there it is in the cytoplasm. The cytoplasm in this figure is a yellow background. So think about it like a cellular broth, okay? In that cellular broth, we're going to have some structures. We have plasmids, which are additional circular pieces of DNA that can lend some properties perhaps to the cell. We see inclusions, which look like a little bubble, like maybe from a bubble bath. There we go. Inclusions can have like waste products in them, but they also can have gas, so it can help the bacterium essentially be buoyant. So raising up in a fluid of water where they need more oxygen, or lowering down if they don't like sunlight, or if they do some other kind of metabolism. Okay, so then outside in, so right enclosing that cytoplasm is that plasma membrane. And then we have the cell wall. I don't know why my cursor keeps moving, but there we go. I'll try to keep it here. Then we have our cell wall, and we're going to talk about those cell wall properties in a bit. And then we have a capsule. So this capsule is a thicker layer enclosing the cell, and it's going to have some different properties as well. So we'll come back to that in a minute. Lastly, endospores. So endospores are essentially resistant propagules, or as you might imagine, like a fungus makes a spore. That spore helps it in reproducing itself during tough times. Well, endospores are highly, highly resistant to really unfavorable conditions, and that's how the bacterium is living until conditions get better. So an endospore is a last-ditch effort to make a resistant propagule, or part of the body, if you will, or this reproductive cell that then can become active when conditions get better. Okay, next, I have about 5 minutes in this first video, but the way that we're going to start differentiating between different kinds of bacteria, not only that idea of archaea and bacteria, but we're going to take a look at cell shapes and arrangements. So what is the cell, what is the shape of the individual cell, and what is the arrangement when it's with other cells, okay? So we know that bacteria can live single-celled. They can develop colonies. They can develop biofilms, which can be different species. So essentially that slimy layer inside your dog bowl, or sometimes when I go to clean out like the drain in the shower, there's this orange gunk and it literally kind of peels off in a film. So we are going to talk a little bit more about that. Okay, one of the shape is cossi or singular coccus, which means essentially circular around. So you can see in the far right-hand corner, there's a figure of stained coccus-shaped cells. They are round, but do know that, to be perfectly round is a little not so ideal in nature. So they might look like a corn kernel, like this one here. They might be a little bit oblong, like the shape of a bean. Okay, so what we see in this photo is coccus cells, and they are together in a group that looks more like clusters. So we call that staphylococcus. Staphylo means clusters. Okay, the next cell shape is the rod shape. Singular bacillus. If you see bacilli, that means it's plural. So those cells in the genus Bacillus are in fact all rod-shaped. There are bacillus-shaped cells in other organisms like Escherichia coli. So again, look at these rod shapes. They can look like, I don't know, a piece of rice. They can look a little bit more like a dog bone or a dog biscuit or something. They can look really skinny and long. Or they can look like literally a string of hot dogs, okay? So what we have here are rod-shaped cells. So think longer rather than wide. Okay, the last bacterial shape essentially are our curvy things, either slightly curved, and that's in our genus Vibrio, but also the name of that cell shape is Vibrio. And then we also have literally spiral-shaped bacteria called Spirillum. You will also see the term spirochete. When you see spiro, you think spiral. Chete, they're a little bit more flexible and likely have some things that are going to help them move. Okay, bacterial arrangements. They can be all by themselves, so just a single cell. When you see the prefix di, means two, diplo means pairs. So we can have diplobacillus, just two bacillus cells connected. We can have diplococcus, two coccus cells together. So break down the word. Tetrad means four. So I'm going to show you how that can happen. Staphylo, we talked about being clusters. Staphylo comes from the word, I think that relates to like a cluster of grapes. Strepto, like streptococcus, is a strand that looks like a string of pearls. And then Sarcina or Sarcina is cubical packets. So let's take a look at what this can look like. We have three figures here, so separate them in your mind, A, B, and C. In A, it's essentially showing us that when you have division in one plane, then you're going to get these either diplococcus or you're going to get a strand. Okay, so they're just dividing in one plane. So think about like X, Y, and Z. So in plane X, or actually, I guess that's Y. Okay, in figure B, we have division not only in that Y plane, but the X plane. So now we get them dividing in two directions. So we get our tetrads. Or if those four also divide again, then we're going to get this three-dimensional cube-shaped packet, and it can be 8 to 64 cells. And then in figure C, it shows us that you have divisions in many different planes. And so we get this cluster appearance. So they might just be, 4, they might be 8, they can be more. So it just depends on that particular species. I will start here in our second part. So I will pause and rejoin you in just a moment.